starting with today's journal event. Today's journal event is more specific to the bioinformatics field. We'll be discussing a novel approach called POP-SEQ, which detects protein to RNA interactions. Today our speaker is Neel Sangani. He's an alumni of Luddy as well as the BMI Club. So I'll let you take it away.
Thank you. Thank you for the introduction, Saiti. It's my pleasure to be here. All the formalities that you can think of, I just said. Today's topic, I think, is POP-SEQ, a novel approach for unbiased detection of protein-RNA interaction. It sounds like a jargon right now, but don't worry, we're going to break it down into small pieces. And even if you don't have any background in biology, you will learn something by the end of the talk. Okay, so a little bit about me. I did my bachelor's here at when it was IUPUI back then in biomedical informatics, and I did a minor in biology. And I did my master's thereafter in bioinformatics, and at that time it turned into IU Indianapolis. So I have 2 different degrees from the same school. During my time here, I worked under Dr. Jenga, so Jenga Lab, and majority of the time I worked on RNA biology, which we'll be talking today. Currently, I'm a research analyst in Apostolova lab. She's an MD neurologist who works on like early onset Alzheimer's disease. And so I'm helping her on that area. So it's a neurology department in IU School of Medicine here. So a little bit of background into the talk that I'll start talking about in a sec. So central dogma of biology. Can somebody tell me what central dogma is? Like basic 3 things. We have DNA, transcription, translation. Yeah, DNA, protein, DNA, RNA, and protein. So DNA gets transcribed to RNA, RNA gets translated to protein. All right, that still is a lot of jargon. So to people who doesn't know. So DNA are all genetic information in our body. It holds, it's like a telephone book. So that's like a classic way to explain it. It's like a telephone book of our whole DNA, of how our body looks, how our different parts of our body functions. RNA is a message which is sent to the body to do specific function. So it doesn't really does anything, but it carries the message to the rest of the body. And the proteins are the actual workers of the body, which actually does all the functions and keep our body functional. So in this figure here, the circle we are seeing is the nucleus. And that X-looking thing is a DNA. And it's a chromosome. There are multiple chromosomes. We have 46 total, with X and Y included. It goes through this DNA replication to keep generating DNA. It transcribes into RNA, which has the information. And then the RNA gets shipped out of the nucleus into the cytoplasm. And there, messenger RNA comes in contact with proteins like ribosomes to turn these RNA transcripts into the protein, functional proteins, which does downstream functions. Okay, so I think future slides here, we go into much more detail. So we know central dogma now, DNA, RNA, and protein. Now, for these to function, there is something called RBPs, otherwise known as RNA binding proteins, right? These are the guys who are like, I would say, project managers. They don't themselves do any like actual like metabolic functions, but they are instructing RNAs to do specific functions. So there are like 5 different things here that's shown in the figure, but they do a lot more than that. So one of the things is splicing. So splicing, when messenger RNA is in so it's not even messenger RNA, it's like pre-messenger RNA. Because messenger RNA is when it leaves the cytoplasm, it's already done splicing and leaving. So pre-RNA goes through like 5' capping, polyadenylation, it goes through splicing. So all of this is regulated by RBPs. These project managers are overseeing all these different things happening within this nucleus, saying like, okay, you need to cut here, or you need to add a 5' capping, or you need to make sure the poly A tail is preserved. So when we leave the nucleus, the RNA is not degraded, right? So we need to understand which RBPs, which these project managers are, and where are they binding, and what are they doing now? Which splicing are they doing? right? So we need to understand who these project managers are. And so that's today's topic is. I'm going to be talking about a lot about RBP. Who are these guys and where are they binding? Okay. So another example is RNA transport, right? So these guys, you can see in the figure, this HUR. HUR is a protein, RNA binding protein, RBP. It's binding to this RNA and then allowing this to move out. And then it helps you like go to a specific place. So RNA doesn't just simply leave the nucleus and just go out in a cytoplasm. It has a specific designated spot where it needs to be. So this RBP, when it attaches to the RNA, it goes and travels to a specific spot, and the set of ribosomes in that area then starts doing its translation, right? So it can't just be dumped out in the nucleosome. So it needs direction. The direction is given by the RBPs, right? Like a project manager. Okay, RNA localization, that's similar to transportation. Transportation allows you to get out. Localization is where do you need to be, and it interacts with this. You can see the cytoskeleton, right? This is this gray area has a cytoskeleton, which is like protein, structural proteins, right? Which like holds on to these RNA, binds to the RNA, and makes sure this whole mechanism stays in place. So RNA binding proteins has two things. So it has a domain where it binds to the RNA, right? And it has an auxiliary domain, which it binds to another proteins. So it binds to the RNA and it binds to other proteins, and it acts as an anchor, right? And so this is kind of what we're seeing in this case, because it's binding to the RNA and it's binding to this whole cytokeratin, right? Acting as an anchor, and it's allowing this RNA to do whatever it needs to do in this area, right? So it has multiple purposes, and it's essential to learn what these functions are. Fourth is RNA stability. So RNA sometimes doesn't just like start translating as soon as it leaves the nucleus. Sometimes what it does is it's ready, it's pre-processed in the nucleus, and it stays in this like granule in like what's a phased state where it's not actually like immediately translated, but it's like kind of like frozen state in a granule. And that is done through the RNA binding protein because it binds and it tells that, hey, we don't actually need you right now. Like, can you just stay here? And we will activate you once there is a shortage, and we'll start bringing you to the ribosome, and then we'll start making proteins. So we need to study what these RBPs are and how they're like stopping these RNAs from getting translated. So there is also a job for that. So the RNA stability plays a major role as well in these project managers. Personnel. Okay, then we have translation control. Naturally, ribosomes, right, who are doing this translation. We're seeing these two ribosomes, 80 and 30, right, doing their job. And we have the cap here, M76 modification, the poly-A tail, and then we have this protein, which is telling at what rate should you make this. You shouldn't make it too fast that it breaks off, or too slow that the demand is too high that we're not matching the supply. Right? And so the RBP kind of controls the rate of this translation. So this is kind of the gist of like the power of RBPs and why we need to study them. Because without them, we're just kind of, so back in the time when research was done, we were just looking at like the physical change, right, under the microscope. But now these guys, because they control RNAs, we're seeing gene expression changes. Like we have overexpressions and underexpressions. That's where the NGS comes in, microRNA, and we have this next generation sequencing, and now like we have Oxford nanopore, right? So all these technologies only came to be because we saw a change in gene expression, and these are the major controllers of gene expressions because they bind and they increase or suppress gene expressions, right? So to get into this talk, so the whole aim of the talk that I'll give is the transcription-wide global protein occupancy. The capture of transcription-wide global protein occupancy using novel POP-SEQ method. So when I say capture of transcriptome-wide, I mean like everything from like within nucleus to outside the nucleus. There are studies which only studies like specific area of cell, and it doesn't study the whole transcriptome. So I will talk about the different studies as well, and then why this is global. And the global protein occupancy is that all different types of proteins. So what different kinds of proteins do you guys know? Name a few from biology. Different types of protein. We talked about splicing. What types of proteins are involved in splicing? Chaperones? Helicase is a mechanism. Or do we have a protein? Histone proteins, right? RNA histones. Yeah. Spliceosome, transfer RNAs, right? What else? Link RNAs. Have you heard of link RNAs? Long non-coding RNAs? Ligases. Ligases, right? MicroRNAs. Have you heard of microRNAs? We just got Nobel Prize in the study of microRNAs. I don't know if you've heard of it. It's very recent. Then there is snow RNA, small non-nuclear RNA. There are multiple types of RNAs that needs to be studied. And all these RNAs are interacting with these project managers, RNA binding protein, right? And so where are they binding and how are they playing role is a massive knowledge gap. It's not even studied yet. So that's when the postdoc that I worked with decided to make this protocol and we kind of analyzed. So we're going to break this down a little bit further. So to detect these RNA binding proteins, there were two different areas where they started studying. So one is protein centric. So we would have a protein which is binding to RNA, and again, we're interested in this guy and where this is binding, right? So what these guys did was they were like, okay, let's make an antibody. This is engineered antibody that they would create in a lab, which is complementary to one region of this protein. They're like, okay, we're gonna bind antibodies to these protein of interest, and then wash everything else, and all the things that is binded to this antibody, we're gonna only take those out, and then wash this protein and study this guy, right? So that was one set of group who was studying this. Other set of group, they were like, okay, we're gonna strengthen this bond here, which is RNA, a protein binding to RNA. We're gonna expose UV light, formaldehyde, and we're gonna really strengthen this guy, and then remove everything else through washing it. So they would wash away everything else and only pick this guy through cross-linking. And they would have a poly-A oligonucleotide that they would make a complementary tail, and then they would do a pull down. They're like, okay, only these guys with poly-A tail and this cross-linking will only get pulled down. Or they would make a magnet for this, and they would be pulled down. And so there was another set of group. So we kind of fall under RNA-centric group than protein-centric. But I'll tell you different types of biases that each approaches have, and then why we decided what we decided to go with. So the first one is RIPSEQ, which is RNA immunoprecipitation. So it directly uses the antibody like it shows in the photo. It has a direct and indirect interaction. So sometimes the antibodies that are designed, they don't necessarily only bind to the protein that we want them to. They bind to random protein. And then when we do the pull down, we think it's a false positive, right? We think that, oh, this is a pull down, this protein must be binding to the protein of interest, RNA of interest, but they're not, because there is a false positive. This antibody is also binding to some random random protein, and then we're doing the pull down on that. So there is a false positive on that. So there is another method called HITS clip, which is high throughput sequencing of RNA isolation by cross-linking immunoprecipitation. I know, a mouthful. But these guys, this is when the next generation sequencing and everything started to take off. And they were like, okay, we need to leverage microRNA and next generation sequencing to further detect specific targets, RBPs. And that's when they introduced this covalent bond using the UV radiation. And then they would do the pull down based on proteins. Right, but the UV cross-linking also was non-specific, because when you introduce UV cross-linking, you can't really target one spot, because it's going to do radiation on everything. What about every nearby places? It's going to, when you do a radiation, it's not like you're gonna add a specific scope and only do radiation on one thing. You're doing on the whole culture. So when you do that, you're also doing non-specific nearby interaction. You're strengthening those bonds which are not actually strong. And when you do the analysis, bioinformatic analysis, it shows as if they have a really strong bond, but it doesn't have strong bonds, right? And then lastly, we have this PAR clip, which is photon ribonucleoside enhanced clip, which they use a different kind of photon. And then there is POP-SEQ, which is similar to what we're doing here, protein occupancy profiling, but they use this photoreactive nucleoside, meaning that we have in a sugar, we have a side. So we have ATGC or adenine thymine. So all of them, what they do is they add this photoreactive nucleoside on the side, and then they do pull down based on that, because in our RNA we all have a modification at the end, and we can do pull down from that. So that's what they were utilizing. But that also have a lot of biases and a lot of offset effects. And then oligo DT capture, that's the poly A stuff that I was saying earlier. Like they make so it's AAA, so they make TTTTT, and then they may complement that and then pull it based on that. But the limitation is that we're only studying RNAs which has poly A tail. And we talked about earlier, right? What about histones? What about like link RNAs or microRNAs? So all those guys, we're not even studying them because we're only doing poly A pull down. So there is a massive gap of studying all these different types of protein. We're only studying like very targeted ones. So what about the others? And that's when like we came up with this protocol, right? Protein occupancy profile sequencing. So in this guy, we use this triazole-based phase separation. So we have these protein interaction happening, right? Both inside and outside. In the nucleus, outside the nucleus. And you can see two different types of protein, right? That's like, say, one's black, one's like orange, brown, and then one is green, right? It's happening in two different places. So what we do first is that we break down this barrier. So everything is in the one pool. And then we add this triazole interface. So we literally just do this interface where triazole allows them to break down into different phases: aqueous phase, inert phase, and organic phase. So organic phase is like any floating RNAs, they just get, because they're heavy, right? Proteins are usually heavy by nature because of carbon and all bonds. And so they settle down. All the aqueous phase, which are like all the like free RNAs, which are not bound to RNAs, they start to float up. And any like bounded RNAs are then settled in the middle interface layer. So if you notice in this example, in this experimental protocol, we didn't use any UV radiation. We didn't use any formaldehyde, which adds biases. So we didn't do any of that. So in that case, it's very pure in detecting all these interactions and the level of strength they have. Second thing is we're studying protein RNA within the nucleus as well, not only outside, but also within the nucleus. So that was a plus. And so naturally, we're going to isolate the interface. So we just take out the aqueous phase, we take out the organic phase, we isolate the interface, and then we do the RNA digestion. So we break down all these RNAs into only the places, the domains where the RNA and the RNA and the protein are binding. And then we remove the proteins and only keep the RNA. And that's where the proteinase K digestion comes, right? So we're like digesting all the proteins, only keeping the RNA fragments, and then we sequence them. So it is RNA depletion, and then we do the sequencing. So we have about 50 to 70 base pair long RNA sequencing fragments where these proteins are binding. And we're interested in this region where these proteins are binding. Now, what questions do you have after learning this? What do you guys can think of? So we only have RNA, like these RNA fragments, and we don't know which protein because we digested them. And it's not like we're only studying one protein. We're studying multiple proteins inside and outside, right? So how do we know which protein bounded to which RNA, right? And how do we know like what was the strength of it? Any ideas? How do you guys think, like how would we study them? Like intensity of it, for example. Like suppose this blue protein, right? So how strongly it's binding to that RNA? How would we study that? Right? So it's not like we can have a strength test or anything, right? So the only thing available to us is the fragments, RNA fragments. So the one thing we can think of is the number of RNA fragments. The more RNA fragments there are, we can tell that, oh, this protein is frequently binding to this RNA fragments that we have multiple of the same protein. So we have a lot of this one specific type of RNA compared to this orange fragment, which is binding only a few times. The number of RNA fragments tells us how strongly they're binding, right? Because their binding is so frequent. And because they bind for, if they have a very strong bond, then when we do this wash, there's a wash between here, right? So they don't get unbounded because when you're doing wash, it's a physical wash. You're like shaking up the RNA and protein. If they don't have a strong bond, they will like break apart. And so it wouldn't end up being here because they will get washed away before you even get to the digestion step, right? So things like this kind of helps think of what ways we can study these RNA fragments, right? Okay, what other questions do you have on this slide? We can talk about it, I can expand it that you guys didn't understand. So do you guys understand that there are two different types of detecting RNA binding proteins? One is like antibody specific and one is like RNA centric, right? And you understand why this is RNA centric because there is no external antibodies or anything exposed. There is no UV light or formaldehyde or anything added to this. And it's very pure if you think about it. Like, it's not as complex. It's a very simple method. So we did this and we expanded the study. We first studied like using cross-linking and non-cross-linking. So we did another experiment where we did introduce UV cross-linking. To see like if it's actually better to add UV cross-linking without this, and we saw like 70% of the overlap. So then we can say that, oh, without UV cross-linking, we're still getting 70%. So doing this is better because we're confident that we're not adding biases, right? So we did that, and there's several other analyses that we did with this. So, okay, any other questions with this? Yes.
So when you were talking about the phase separation, I'm guessing you take out.
Pipette?
Yeah, the pipette, and you take out the organic phase.
Yeah.
So how?
Organic phase.
Yeah, so how confident are you, or like experimentalists, in just isolating that one patient? Like, what if the pipette accidentally takes out the interphase region?
There is a risk. Yes, absolutely. That you can definitely do that. And they have to go through multiple washes. It's not like you just do one wash. Like you add isolates like multiple times to make sure that the separation is proper.
Right?
Yeah.
Serial dilution.
Yeah. Like you have to, do the multiple dilutions, but make sure that the interface stays intact. I'm sure like some part of it you did take out because human error, right? It's very hard to not do that.
Well, it's depending on when you prove them for really fresh, we already take the histology or the type of preserve them for.
You're saying like after creating this, like freezing them
Yeah,
and after like you reusing them at some point.
yeah.
Yeah.
Maybe one time thing.
This is very, you have to use it in French. It's very like time constraint. You have to, as soon as they're like, because if you don't do that, then this interaction will start getting weakened and then you won't be able to isolate the RNAs, So, this is very like timed and you have to like do these in specific like time frames to make sure you get all the interaction. And I was giving a talk in Michigan, right? And I was talking with other people who were working in this area. And apparently like this step is what everybody does. But they never came to think that they can just use interface and extract all the protein-RNA interactions and study those. They would always come to this step and they'll add like UV radiation or formaldehyde, like I talked earlier, all these different methods. But they never thought of like just using interface and then extracting them and studying those.
It's interesting just because in so many other fields of biology, it's a very common way to extract things.
Yeah.
You put in a centrifuge, then you just
Yeah.
pull out the top.
Yeah, so everybody did that. And when I was presenting this poster, they were like, what, you can do this? And these were other postdocs in other labs who were experimentalists. And they were coming up to me saying, like, you can do this? What? We've been just throwing it away as if it's nothing all this time. And like, this is the first time that anybody has told us that we can do this. So that was exciting to know that actually like this part was very novel and like nobody thought of this. Yeah, so we did that and then this is one of many cell lines that we analyzed, and this was part of my thesis, and so I'm kind of presenting this. So K562 and Zircon are two leukemia cell lines. So we just finished what? We did this RNA, we got this RNA digestion, and then we like depleted rRNA. So what is rRNA? Ribosomal RNA. Why don't we need this in our data? Yeah, because remember? Yeah, they're everywhere, right? They're the ribosomes who are making RNAs into protein. So they're everywhere, and we don't need them. Like we already know what the 90 and 30s are, what they do and everything. So we don't need RNAs. So we have to deplete them, and we also need a kind of similar study, which is like we download data from open source, and we saw like cross-check to make sure we don't have any noise, because we have to make sure that majority of the data that we're saying like, oh, we found all these RNAs, but what if majority of them are all rRNA? That would be a waste, right? Because that's all noise. We don't need that. And so rRNA depletion is important because we want to study other RNAs. And then these are all modifications for like analyzers, so we can get the small reads. Okay, now question is, how many of you guys know next generation sequencing, microarray, Sangani? Who is not aware of it? So not aware, okay, no worries, then we'll go over it. So what happens is we have these RNAs, and RNAs, I don't know if I want to go through the whole like detailed version of it, but I'll give you like very tiny. So like these RNA molecules they have like these primers on the front, and the whole machine this bioanalyzer, these QC things do is they just make multiple copies of it. And so instead of this one RNA, there's like multiple of it. So it helps us analyze it better, right? So it turns it into, so this is a physical biological like RNA, and then it reads the signal into like ATGC, adenine, thymine, guanine, cytosine, right? And it turns it into this huge massive file, FastQC file, and then you can QC them and all. But that's what the short read sequencing Illumina sequencer is doing. It's one of the sequencers. There are multiple sequencers out there. It's doing. So you have this massive file, could be like GBs worth of data, and then you sequence them essentially, and to figure out the trends. And we can kind of go over it as I go. So these two cell lines that I'll talk about, right? So quick background of these two. So these are chronic myelogenous leukemia, which is K562. It's derived from a 53-year-old female. I don't know if you guys know about her, like, no, this is the one about it. I was thinking of HeLa cell line, which was different cell line, Henrietta Lacks. But 53-year-old female, that's where the K562 was derived from. And the T-cell acute lymphoma, lymphoblastic leukemia was derived from a 14-year-old boy. So these are both leukemia, but one is for 53, which is like an adult, right? And then 14 is like a young. So we're trying to see that what are some of the genes which are common between both of them. And they're both leukemia cell lines. So in this study, we're interested in that. And they both are essential in studying like T-cell signaling and cancer research essentially. So we have a lot of research done in this area, and these two cell lines are very frequently used to study like molecular mechanisms of like what different specific proteins are doing what biological functions, or therapeutic targets as well as some of the vaccines or some of the drugs that came out. They all studied these leukemia cell lines to come up with that. Okay, so this is the workflow of the two cell lines. So Jurkat cell exhibits high proportion of peak in intronic and K562 cell lines in exonic regions, and we'll go over that. So quickly over the flow line. So we have POP-SEQ reads. So this is the Illumina sequencer giving all these reads. Then we do quality check. Then we do alignment. Then we do peak calling and annotation. So we know what reads are. So reads are, again, I said ATGC is a line of 50 to 70 base pair long. And there's like multiple, millions of them in a file. FastQC, what we're working on here is adapters. When we do the QC through the Illumina sequencer, we add adapters in front of these reads to help them do the PCR, right? What's the full form of PCR? Chain reaction PCR, yeah, so we do PCR and we use adapters to do this, right? And so sometimes these adapters are not trimmed. The Illumina sequencers are supposed to trim, but they are not always trimmed. And so what we do in QC is we make sure that the data that we're using is actually like human data and the quality is good. There is math behind it. They look for like 30 and above, 32 and above. For quality check, we do specific error rates, 1 over like 100 or 1,000 or something. And then adapters, if adapters are not removed, it'll tell you like, oh, there's adapter contamination. And then you would go back and trim like front of it. And there are different types of reads too, by the way. There is a single read and paired end read. I think ours was single read. It wasn't paired end. Paired end is like they read from both sides. So 3' to 5' and 5' to 3', both ways. And it gives more confidence because they're overlapping. And then alignment is when we are aligning all these reads to the genome, actual human genome. And again, these are all human cell lines, right? So we're using human genome, and there are other genomes too, mice genome, zebrafish genome, any like which are like compiled genomes. So we align it to that, and it tells us the quality of these reads, so certain percentages. But finally, all these reads that do come out after alignments, we know that they do exist in the alignment. After that, now we're interested in the question that I posed earlier. What are the RNA binding proteins? Who are they? Who bind it to these RNA fragments? Right? We don't know because there are so many RNA binding proteins in the cell. And so we have all these reads, they're all like together. And we don't know which RNA binding protein bound to where and how much, right? And so then we're doing what we call peak calling, where we're figuring out like the intensity of these reads. We're figuring out where they're binding. This is analysis. Piranha is like a tool. So anything that's written below, these are all like specific tools, bioinformatics tools that I use to analyze these. Okay, so HiSat tool is a specific tool. There are multiple aligners. There's top head, there's bow tie, there's so many different types of aligners. So I use HiSat 2, and then the GRCH38P, 13P that is like a specific version of it. It's a human genome. And you align it to that, and then you get all these reads. After you get those reads, then you look for peak calling, which is where the RNA, this protein is binding to the RNA, so the domain. We're interested in the specific domain of these RNA. Because RNA fragments could be 70, base pair 50 to 70, but the domain could be like from 5 to 12 base pair. So from 70 to 5 to 12, it's like a specific region, right? So we're interested in, and that's what peak calling allows us to do. What do they do in the background? They essentially take multiple reads and they run this Gaussian model to like figure out multiple overlapping peaks which are very frequent. And then they say like, okay, this region of the reads are very frequent and are also seen in our databases that a lot of the protein have the similar domain. And so these domains are found in a protein. Very technical side, but you don't have to worry about it. But that's what they essentially do. And then the Homer is, it tells us like what different types of protein RNAs are found. And so when this protein are analyzed, we are interested in like what different RNAs are found. Are they intronic, exonic? Are they linked RNAs, no RNA, right? What different kinds of RNAs are found? And that's what the peak annotations allows us to do. And the tool I used is Homer. So, okay. So kind of going a quick overview. So how many total thousands of peak? I'm going to go here. So we have 2 replicates for each cell line, and we have control for them as well. So which is not included here. But we have about what 120, 130. This is in thousands. This total peaks. They don't have reads because this peak, this first figure, is showing from like Piranha peak column. So alignment is done. And now we do the peak calling. So we're seeing like about 120,000 peaks, which shows that we have that many interactions with proteins. 120,000 interactions with RNA. Say for both cell line. For Jerka, we're seeing like over 300,000. right? Over 300,000 interactions, so over 300,000 RNA proteins are binding to the RNA. That's what we're finding here. And then we do the combination of them, combine them together into consensus to make sure that these interactions we found in individual replicates are actually like true. It's not like two different players are playing a role. So we combine them, like we didn't combine them, but we overlap them to make sure that we are seeing the same players in both replicates. So we're confident. Same thing goes with both cell lines. And then this is more of saying that the read depth, that we did the same for both, but we clearly see like there is more read depth in Jerka. So when we do PCR, right? While we're generating reads in Illumina sequencer. Some of them generates more reads, some of them generates less reads. So there is definitely some biases towards Jerka. It could be because naturally they have more data maybe, or they are more prone to like replicating faster when you do PCR. It could be various reasons. So finally, we took this peak now, consensus peak, and then we did the annotation. So we know that there are about what, 80,000 peaks and then 30,000 peaks. So we can see we dropped, there's a major drop from here. We saw about 120 million, 300,000 to 300,000 peaks to 25,000 and 85,000, right? There's a massive drop. Why did that drop happen? Because we're only looking for the common players between both replicates, because we want to be confident that these players are found in both replicates and they're not just like noise, right? So we have this, so we have 85,000 RNA binding proteins binding to the RNA. That's what we know so far. But where are they binding? We don't know. We just know that this many players are interacting. And this is transcriptome Y. So the game is crazy because we have so many RNA binding proteins interacting with RNA. So now we're interested in where are they binding? We don't know who are they. We haven't even gotten to that question, right? We're first asking where are they binding? So they're binding to intron, promoter, exon, 3' UTR, and there are multiple others. No RNA, that's like siRNA, microRNA, link RNA. There's so many of them, but they are less than this percent. And again, this is proportional consensus peak. So this is not total number, this is percentage of the total amount, right? So majority of the Jerk cell lines, we're seeing that have more intronic activity over POP-SEQ. So essentially we just saw, so I don't want to get into jargon of this, but want you to know that we had a protein which was binding to RNAs. We didn't know where it was, and now we figured out where it's binding, right? So now we know where it's binding. The question now next is, who are these players, right? Who is binding to intron? Who is binding to promoter? Who is binding to exon, right? We only know RBPs are binding, but which RBP is binding? We don't know that. So that's the next question, actually, right? So I don't want to get into too much details of jargon, but I want you to think about like where we're heading, right? Okay, so now next is, so we did peak calling. So we have all these, right, protein-RNA interactions. Then we did differential peak. Differential peak is now we're comparing these two players, K562 and Jerk, saying which one has more stronger binding compared to the other, stronger affinity over the other. So we have K562 at the top and Jerk at the bottom. And so we can clearly see from this visual perception that we're seeing more dots on this side, right? So Jerk has more stronger binding compared to K562. So that tells us something that, oh, Jerk is a major player compared to K562. And Jerk is what, a 15-year-old, younger, in younger cell lines, female cell lines versus older. So there could be some players. We can study that further. And then we look at number of peaks. So how many number of differential peaks? So differential peaks, they look at like only stronger affinity. So all the like normal binding, it's usually filtered out. So this is usually filtered through like log change and FDR. So all that filter adds on. And then we like filter down even further. So if you remember, it was like 25,000 and 85,000 RNA binding proteins. And now we're down to 12,000 and 13,000. So 13,000 is specific to lincRNA. It's a subset of this. So lincRNA also play a major role because they're non-coding RNAs, right? Non-coding RNAs bind to RNA directly and they can silence them. So microRNA, for example, right? There are about 12 to 15 base pair long, and they bind to the RNA and they can like stop and ask them to like die immediately. And so we need to be aware of where they are and what they're doing, because there are a lot of now pharma companies which are designing these microRNAs so they can silence the RNAs which are causing downstream effect, any like diseases, if they're like triggering overexpression of specific molecular functions, which is causing like production of more protein, which is causing damage, right? And so these microRNAs goes and bind to those messenger RNAs and stop them completely from doing that damage. And so it's an interesting study. And so these project managers are also binding to lincRNAs and microRNAs and regulating them. So we're also interested in studying that as well. And after doing the differential, so only the strong RNA binding protein interactions between both cell lines. Then we did that this again using Homer, right? To see where they're also binding. So we also see like other very different players who are playing a role. Yeah, I think that's good for this slide. Any questions? Yeah.
Is it binding more to do with sample?
No, this is affinity. So how strongly are they binding? So like I said with reads, right? So if it's binding multiple times to the same reads, we will see a lot of those reads in the sample. And a lot of those reads shows that, oh, hey, for the same RNA binding protein, we're seeing way too many reads for this cell line versus not as many for the other cell line. You can infer that, hey, like this RNA binding protein must be binding more to this cell line compared to the other. Therefore, must be regulating more from this cell line compared to the other. If it's regulating more, then it should be more of an interest because it has a stronger bond, a stronger control for one cell line. Any other questions? Okay, so now we have this list of, peaks, right? So this list of peaks are essentially RNAs. RNAs where the proteins are binding. So these RNAs must be a gene, right? This RNA should be a gene, essentially. Every RNA is part of some gene, unless it's a lincRNA or microRNA. They should be part of the gene. So then we use this GTF file, genome annotation file, to find which gene these peaks are associated with. So we get a whole list of gene lists. So whole 12,106 gene lists, and some of them could be overlapping too, right? There could be a lot of replicates, a lot of repeats, and so we'll filter those and all. But we get the whole list, and then we ask question is, how are they all related? What kind of functional processes we are seeing when we group all these genes together? And that's what we do, this functional enrichment analysis. This was done using Klugo, if anyone is interested. But essentially, this gives you this whole like network of different pathways. They're all associated. So this circle here has a set of genes which are all connected somehow, which is causing this functional, this function process, right? And all of this, like people have extensively researched these pathways to know that this set of genes together makes this pathway, right? And I'll give you one of the examples in shortly. But this functional pathway kind of gives you the idea of like what different types of like functional processes are involved in a specific cell line. So we're looking at leukemia. Now, as an experimentalist, you should be looking for leukemia-specific pathways, right? You would be like, okay, this is a leukemia cell line, and we have all these genes now from the peaks that we found, which are the RNA binding proteins are binding to. We should be looking at like microRNA analysis or like some sort of immune response related pathways, because this is a leukemia. Leukemia should be a T cell specific stuff. So they will be looking for that, and all these RNA RBPs should be binding to these genes to regulate them. And so this is a way for them to kind of confirm that the data that they have generated is not just noise. The actual real data, and we can actually trust and further study and go beyond like these RNA binding proteins, and now we can actually like go very specific, right? So these are the ways for them to kind of confirm that we're studying the real thing. So there's a lot of different things. So here's microRNA is one of the things. There's a cellular response to stress, protein processes, right? Protein modification, tRNA we talked about. So all of these are found in these pathways, which kind of says that, hey, the global analysis that we were doing it's there. We're seeing all these processes. And then there's all these other ones as well. So that was specific to K562. And then the other functional analysis we did was for jercut. And jercut you see is now here on DNA level. We have a DNA binding, chromatin organization, chromatin, right? So this is within the nucleus. And then the last one we saw was outside the nucleus. So this also tells us a lot. The jercut is controlling more like transcriptional level, whereas K562 is post-transcriptional level. Because you can see, right, there is tRNA, which is what protein amino acetylation, protein translation, right? Protein modification, which is all happens in cytoplasm. So these key processes kind of gives you an idea of like which area you should focus more on. I don't know why this is still blurry. Okay, so the example that I wanted to tell you, before I get to that, I want to show you this. So MTR gene, this example is showing the gene expression between two cell lines. So the top is jercut. We have two replicates, replicate 1, replicate 2, and we have K562, replicate 1, replicate 2. And this is the total RNA, which just shows like the expression of them. So they all are equal expression. So there is no bias in that sense. And we can clearly see that jercut has more binding compared to K562. You can see there is more higher expression. Again, how do we see higher expression? Because there are more reads, right, in that area for this specific gene. So this is an MTR3 gene. And what other interesting thing is there is this SNORA74D, which is a SNO RNA. This is a non-coding RNA. This is a new, like it's very small, tiny, micro. I don't know. It's a SNO RNA. It's not a micro RNA. It's a SNO RNA. It's a non-coding RNA which binds to the RNA and could play a role. So again, like I said in microRNA, it binds. This is a non-coding RNA which goes and binds to the RNA, and it could suppress its function. And so having that right next to the gene that we are noticing here could play a lot. Like we can literally design that. Like a pharma company could design that, and we can control this guys, like how they're expressed, right? That could be of interest. So just knowing that we found that, that's pretty exciting. And there is also this another SNHG4, which is a long non-coding RNA. You can see like how long that is, right? It's like a really long non-coding RNA, which also happened to be around. So this is upstream, I would say, right? Because the gene is going that way, 5' to 3'. So this is like a little bit upstream. So upstream, it has a location where this project manager RNA binding protein can come and bind to this long non-coding RNA and can affect how these guys are regulated, whether that is upstream or downstream, right? So there are all these different ways you can regulate this gene, and this gene could play a major role in whatever downstream functional processes, right? So now going back to this, why this gene is important, right? So MTR3 gene interacts with link RNA to upregulate T cell leukemia progression. So we have this link, and it stops the degradation of MTR by this PKA, protein kinase A, right? So when this link RNA is around, like it's nearby, it like stops the degradation of this. Like it doesn't allow, like it doesn't allow the MTR to die. And because this doesn't die, it increases the CCR9. And then the CCR9 promotes the leukemia infiltration, which is so when we say leukemia infiltration, that's when metastasis starts, right? It starts moving to different body parts, right? And so that's what it's doing. So if this dies, then this doesn't happen. But this is not dying because this is controlling this. And this is regulated by our project manager. RNA binding protein. So this is, if RNA binding protein is regulating this is not allowing for this to die down. And this is not dying, and this is, because of that, this is infiltrating now, right? It's promoting leukemia. So you see how complex it is, but how this project managers comes into play and control this whole downstream process. So that's essentially what I'm studying, what I did during my master's here. That I studied all these RNA binding proteins, where they bind, and what their downstream functional analysis processes are, and how to control them. And you don't need to know for every single RNA binding protein. You just need to know a few of them, because a few of them is all it takes to make a life-changing drug discovery. So finding these RNA binding proteins, which regulates these guys, is the whole premise of the research. And so do you guys kind of get the idea of why RNA binding protein is important, and kind of why regulating them and understanding them is so important? Because back in the time, people only studied one RNA binding protein at a time. So they designed a antibody for one protein, and then they would have a whole sample for only one protein. But they have been doing it for years, and so they have generated so much data. So we can leverage that data to figure out which RNA binding proteins are binding to specific RNAs. And then we can start narrowing down these pathways, which changes a lot. So we talked about that. Now going to the motif part. So we know these RNAs are binding to, with this RNA binding proteins that are binding to the RNA. We know which these RNAs are, right? We talked about intron, exons, length RNAs, all these different guys. Now we don't know which proteins are these, which project managers are they. Now it's time to answer that question. Who are these guys? And so for that, we do this differential motif analysis, where we have a database of already people that have studied individual motifs, individual experiments, and compile them into one database. And so we're leveraging that to see that where these domain, right, motif domain that we found across references. So if there is a high hit of it, then we say that we're confident that it is SRSF1 protein for this specific domain, and then so on and so forth. So we have all these proteins with specific significance value for that specific RNA. And that's how we kind of narrow it down. And so you can see this SRSF1 is linked with disruption of tumor suppression pathway, right? We immediately found out these set of RNAs that we have are regulated by these RNA binding proteins, and they're doing specific functions. So we can further explore that and answer some questions. But that's kind of what I want to present today. We can do a quick conclusion, but before that, any questions in this area, at least? So motif area. So we have about, I think, 1,200 motifs, known 1,200 motifs so far. Yeah, questions? We have about 1,200 motifs. We're using, I mean, I'm sure people are doing actively 1 RBP at a time, they're increasing the database. But we can leverage these 1,200 RBPs and find out where they're binding in one experiment. Because we just did one POP-SEQ, one experiment on KY62, one experiment on Zerkat. And we generated this massive file on only just 2 experiments. And we're able to study so many RBPs. Whereas that, like other protocols, this is one experiment, this is one experiment, this is one experiment, right? So we literally cut so much time, cost, and time from research, like scientists to study all these without, spending individual time on each RNA binding proteins on each cell lines. Imagine how many. There's so many cell lines, and studying each RNA binding proteins on each cell line takes so much time. And so this really narrows down, because you can see SRSF1 has 119 counts. You can easily say that, hey, SRSF1 should be our highest priority because they are the major players who are interacting actively in this cell line. And let's just do further study on one protein. But we're only able to do that because we have this whole catalog, right? If we didn't have this catalog, people would be doing for every single RNA binding protein. Imagine you are an experimentalist and you pick this guy and then you only have 55 hits. And you're like, I just wasted my whole week, 2 weeks to culture a cell to study only that. And then it didn't even like, wasn't as effective, right? So it really helps in that sense to filter down and only narrow down to which players we should be studying. And it really fastens our like research to which functional processes we should be like targeting down the line. But yeah, that's kind of, any questions? Yeah. We should change, yeah, essentially, because this SRSF one, this project manager, like, it's only specific to this cell line. They, in completely different cell lines, you'll see completely different set of players who are playing, who are interacting, right? They wouldn't be the same people controlling everywhere. This is leukemia, right? So in other cancer cell lines, you would see other players who are playing. But this saves a lot of time to narrow down the list, right? So you're like, oh, we already know like these top two players, the SRSF1 and FMR1. It's actively binding to everything and regulating everything. So now the question is like, what are they regulating? Now they can be really specific, like these two players, where are they specifically regulating and how are they affecting? Right, so we narrowed down the question a lot compared to like doing one thing at a time, which people do. Some of the labs still do, I'm pretty sure. Okay, so kind of giving you an overview now that this is a trazole-based phase separation method. We do not use poly-A pull down, so it's poly-A independent pull down, and then we have multiple differentially enriched sites with confidence, which again shows future implications in primary tissues. And so this is primary tissues versus immortalized cancer cell line. This is an immortalized cancer cell line. Do you guys not understand the difference between these two?
Yeah.
Right?
Yeah, more or less cancer cell line is from someone that's already
Yeah.
died and should be, taking their cell, they're still living.
Replicating, yeah. So I think the whole motion started, I think it was way before the Henrietta Lacks. But when somebody dies and we have the cancer cell lines, we have to keep feeding resources for them to continue replicating, right? And so these cell lines were done on immortalized cancer cell lines. So we are continuously feeding some sort of food for these cell lines to continue replicating. If we stop giving them the food, they'll die. There was this one cell line, this exciting fact that HeLa cell line, Henrietta Lacks, was the person who died. I can't remember the exact year. It was like 80 something, 90s, 80 or 80 something. But she was the first like person who's like HeLa cell line. She died, and then her cells didn't need any food, just continuously replicated, which is still a medical mystery because it doesn't need food; it just continuously replicates. Yeah.
There's a documentary called Modern Times, The Way of All Flesh from 1997. That's all about Rita.
Yeah, it's so exciting that there is a cell line like that exists, which doesn't even need for any just replicates. But this is this cell line that again I told you, 53-year-old and what 13, 15-year-old. They when they passed away, they took the cells from them and they keep replicating, and that's what the research was all done on, and that's what majority of the research is done on. Before we take it on primary tissue. Primary tissue is like real human person. We're taking their cell and experimenting, and then we're giving them a drug to suppress or express specific, functions. So that's the next step, and hence the second bullet point, right? The future implications in primary tissues. Showing the strength of this differential enriched sites. We're confident that if we take it to primary tissues in humans at one point, I think this experimental protocol will survive. And then it's first of a kind because there is no other such database has been compiled. So this, I only showed you two, but we extended it into six different cell lines. And so it's like a massive database. And it's the first of its kind, and nobody has done this before. And so we compiled that. And then future work. So this is exciting now. I know I'm kind of going over time here, but I'll be quick. So we just did like this whole analysis for two cell lines, Zircon and KO62. Now we can build the machine learning models on them. So we know that these RNA binding proteins are binding to these RNAs. So we know these RNAs and these proteins. We can use these RNAs and these protein informations to predict in a cell line that we have never studied before. So they don't have to do an experiment on it. So the experiment, we're taking even further time away from them. So we take, like, say, like A549, some random, like, lung, cell line, which we haven't even studied. Haven't done the experiment protocol, we haven't applied POP-SEQ on it, right? And so, or actually, no, we do apply POP-SEQ on it, but we don't know what RNAs it's binding to. Right, so it's like a completely unknown RNA set, which don't know which proteins are binding to. So we then can build a protein, this machine learning model where we predict in a completely new cell line, like which proteins would bind in this cell line. And then that would predict, like, based on this domain is binding and based on what RNAs it's binding in this cell line. It'll start predicting in your new cell line, completely new cell line. And it saves you time from even like physically doing them, but before you even physically do them, you can narrow down cell lines down. We first narrowed down the RNA binding proteins, right? Because we had this whole catalog of the refluxer, like SRSF1 had 170 hits, right? So we narrowed down the RNA binding proteins. Now we're narrowing down the cell lines because there's so many cell lines. Each tissues have so many cell lines. Leukemia itself, we just showed 2 cell lines, but it has way more than 2 cell lines. Leukemia has so many cell lines. And so we're literally narrowing down the cell lines using a machine learning model. And that would be the future. Yeah, but that's all for today. Any questions?